Okay. So hemostasis is essentially looking at the clotting of the blood, right? Anyway, sorry, getting distracted. So what we're going to be looking at are the steps involved in clotting of the blood and doing a brief description of the different mechanisms that are also used to reduce the loss of blood, right? So as it mentions here, hemostasis is, of course, the process of halting blood loss. Because, of course, as you can see from the word itself, we have hemo, which, of course, is referring to blood, hemoglobin. And we have stasis, right, which, of course, is referring to the state, right? So you want to keep blood in the same state, basically. So it is the process of halting blood or arresting bleeding from a broken blood vessel, okay? What we have here, and it's very lovely, very important diagram, is I even tick it, is basically breaking down hemostasis into the major steps, okay? So remember, this is a very important diagram. Make sure this one is, you're comfortable with what's going on this one, okay? So it starts off here where you have your vascular injury. Vascular, of course, refers to blood vessels, as I'm sure you all know. You can see here the wall of the blood vessel is torn open. And when you tear open the wall of the blood vessel, it exposes collagen fibers. So what you're seeing here are collagen fibers. Okay. Now this is important. Ordinarily, collagen fibers are not directly exposed to the blood. So when the blood is exposed to them, the blood knows something is wrong because it shouldn't be there, right? Now, so this shows now you get exposed to the collagen fibers that you shouldn't, and this activates platelets floating around inside the bloodstream, all right? And then when the platelets become activated, the platelets then respond to form what is referred to as a platelet plug. And this is called primary hemostasis. And during that process, you also have vasoconstriction occurring behind the break. So blood is flowing in this direction, right? And so vasoconstriction is occurring behind the damaged area. Vasoconstriction, of course, is going to slow the amount of blood entering the damaged area. So you're slowing blood loss. You're also making it easier to form the platelet plug. The purpose of the platelet plug is to pack up into the wound and hopefully stop blood loss. And it is the first thing that happens, which is why it's called primary hemostasis. Primary because it's first. So you don't form a clot to begin with, you form a plug. Remember now, collagen fibers is exposed like in the area of the break, platelets become activated. I'll explain what VWF and all those things are in a minute. Just be aware, you form the platelet plug first. That's primary hemostasis. Now, if primary hemostasis fails, by that I mean even though the platelets are trying their best to block up the hole, blood is still leaking out, that is going to now trigger secondary hemostasis. And secondary hemostasis now is when you get your actual blood clot, right? So you can see here the fibrinogen to fibrin leading to blood clot, right? So remember now, primary hemostasis is platelet plug formation. Platelets packing themselves into the damaged area to try to prevent blood loss. When that fails, you get secondary hemostasis, which is forming a blood clot, which then packs itself into the damaged area. So let's run through the phases again. So the first phase is referred to as the vascular phase. Of course, once again, vascular refers to blood vessels, right? So this is the phase in which you're contracting the blood vessels behind the damaged area to slow that blood loss, right? To keep as much blood in the veins in the blood vessel as possible and make it easier to form the platelet plug. The second step, of course, is going to be a platelet plug formation. And we spoke about that already. So the idea here now is that platelets are going to stick to that exposed collagen and then stick to each other and eventually pack up the space created by the wound and hopefully stop bleeding. Then now, if that doesn't work, you go on to step 3, which is coagulation. This is now when you actually form your blood clot, right? All right, so this is just showing here vascular spasm, as we spoke about before. Now, as it mentions here, vascular spasm now is going to require the endothelial cells input. Now, you all did ANP1, so you should all know that endothelial tissue is lining tissue. Endothelium lines space within the body. The only difference between endothelium and epithelium is that epithelium lines space exposed directly to the outside, and endothelium lines space exposed to the inside. That's really the only difference, right? So as I'm sure you all know, you have simple squamous endothelium inside your blood vessels, right? Now, as this says here, endothelium cells contract from the damaged area and expose the basal lamina to the blood. So the area near the endothelium is damaged. When it gets damaged, the endothelial cells try to pull away from the area that is damaged. And remember, endothelial cells are epithelial cells. All epithelial cells have a basement membrane. You see why it's important to get AMP1 before 2? Because these things that we're expected to know already, right? So since you know that already, it makes life easier, right? So you expose the basal lamina, the basement membrane. And then now that allows now for chemicals to interact with that basal lamina. Then now we have the release of chemicals and hormones from now the endothelial cells. It mentions ADP, adenosine diphosphate. I'm sure everybody knows this one already. ADP plus P equals ATP. That should sound familiar to you, right? You also release substances like tissue factor and prostacyclins, as well as endothelins. Okay, so all of these lovely substances are coming from the endothelial cell in the area of the damage, right? Now, this causes the plasma membranes of these same endothelial cells to become quote unquote sticky. Why is sticky important? Because it makes it easier for platelets to adhere to them. So platelets can stick to the endothelial cells near the damaged area. Platelets can stick to the exposed collagen fibers near the damaged area. And then by sticking to this area, they can build up now and start to block that opening, right? So this here shows the platelet phase. This is the blocking we're talking about now, right? Now, let's talk about BWF here. BWF is known as von Willebrand's factor. The purpose of von Willebrand's factor is to activate platelets and attract them to the area of damage. So here we have an endothelial cell near the damaged area here, right? And the von Willebrand's factor now being released causes the platelets that are floating around to come down to where the von Willebrand's factor is and stick. And of course, you can see the collagen is here as well. So one platelet comes in and sticks. When one platelet comes in and sticks, this platelet then releases other substances from itself, like thromboxane and ADP. And these substances actually recruit more platelets. So basically, that's the phone call. The thromboxane and the ADP is a phone call. So the platelets unite, come down here. So the platelets move now to the area where the activated platelet is, and then they stick to that activated platelet, as you can see here. And then they become activated themselves, and they release their own factors. So this one is doing the same thing, releasing these things, which call more platelets, until eventually they fill in the damaged area. And remember, as long as the damage is not too bad, then you don't need to go further than this. If it's a small hole, this is all you need, right? So here we go again. It mentions that platelets, once they become active, they become spherical and develop cytoplasmic processes. So your platelets are normally sort of flat, right? Kind of like that. And then when they become activated, they sort of look more like this. Oop, you know what? Not like that. That's ugly. Sort of like this. They become spherical and then have protrusions coming off of them like that. Sort of look like a sun. Well, they're not all going to be that long, but you get the idea. And what these protrusions do is that allows them to stick to one another. Go ahead, Tavia.
Excuse me, sir. The factor releases a chemical signal or it is the signal, if you
Factors
understand.
are the signals.
Okay, okay.
So remember now, the platelets go from being flat and boring to sort of being very round and spiky. And the round and spiky platelets can stick to each other more easily, right? And then it mentions here now that the platelets produce various chemicals. It mentions once again ADP. And it says here ADP, remember, platelet aggregation and secretion. So once again, the ADP helps to call the platelets to the area and more of them stick and become activated. They go from being flat and boring to looking like that, right? Here we go, thromboxane, A2 specifically, and serotonin will all increase vascular spasms. So the damaged area, remember the blood vessel just behind the damaged area will constrict, which slows the flow of blood. This makes it easier for the platelets to aggregate and stick. Because if the blood isn't washing them out of the wound, it's easy for them to form a blockage, right? And of course, platelets can also release clotting factors. So if the wound is just too big, the platelets will release clotting factors that will trigger, of course, the third step, right? It also mentions here that platelets can also produce platelet-derived growth factors. And these growth factors increase the rate of mitosis in the surrounding cells. So that, of course, means that the cells around the damaged area replicate more rapidly to quickly seal up the wound. So as you can see, platelets are doing a lot. It mentions here that calcium ions are involved in platelet aggregation as well, and they're important in the clotting process. So as I'm sure you all know, calcium is one of the most important minerals in the body. So this is something else it's involved in, which of course is the clotting of your blood, right? So this is showing you here, this is not a blood clot. This is a platelet plug forming here, right? So these are platelets. Now, if the plug just can't manage, that is when it's time to activate step number 3, the coagulation phase. So vascular phase, if that doesn't work out, platelet phase, if that doesn't work out, coagulation phase is what we have left. That, of course, leads to the clotting of the blood. I want you to understand that your body does not want to form blood clots, right? But you might say to yourself, but a blood clot is the most effective way of stopping bleeding. Why wouldn't you just go for that first? Why not do the most efficient thing first? Here's the problem. Blood clots are like guns. They are useful, but they are dangerous, right? Useful, but dangerous. A blood clot can stop your bleeding. True. If a blood clot gets dislodged from the wound or forms within the blood in the wrong place and gets swept away by the blood, that clot can float around in your body and block important vessels, which of course can lead to things like stroke and pulmonary embolism and heart attacks and all sorts of things. All right. So blood clots are useful, but they are dangerous. So your body only forms them when there's no other choice. So keep that in mind. That's why you don't do blood clot first. You try the plug first because the plug is easy to control. They break apart more easily than the clots. So even if they get dislodged, you don't have to worry about them. Okay? Now, that said, if you have to form the clot, here's how this goes. Clotting is involving three distinct things, or three parts of the process. We have one part that is called extrinsic pathway, then we have an intrinsic pathway, and both of them lead to what is called the common pathway. So extrinsic, intrinsic come together, all lead to the common. Okay? Common pathway is where you actually get the clot forming. These two other pathways are simply how your body activates the formation of that clot, right? Or the common pathway. Now, this lovely diagram here, let me take this one too, is giving you an idea of all the pathways together. Okay? So let us take our extrinsic pathway here. You'll note you start with tissue damage. So once again, the endothelial cell gets damaged, or it's near the damaged area. It releases something called tissue factor 3, all right? Or factor 3 if you prefer. Then the factor 3 has to combine with calcium. So here we're seeing where we're using the calcium. It also has to combine with something called factor 7, clotting factor 7. And these three things, one, two, three, all come together and combine to form factor 7 tissue factor complex. All right? I call it TF7. So tissue factor 7 complex. Right? So your TF7 complex now is going to activate something called factor 10. And factor 10 leads into your common pathway. Okay? So this should be nice and easy to remember. Extrinsic pathway, you get damage to the tissue first. You release factor 3. Factor 3 binds with calcium, and it then binds with factor 7 to give you the TF7 complex. And then that TF7 complex goes on to activate factor 10. All right? So that is all. Real simple, 2 steps. On the other side now, we have the intrinsic pathway over here. Intrinsic pathway, now you notice here it mentions activated proenzymes like factor 12. So we have activated factor 12, and this one now is also can be activated by platelet factor. So between the factor 12 and the platelet factor, we also have to combine once again with calcium, all right, as well as factors 8 and 9. All of them will lead to the activation of the factor 10 complex. Okay? Now, this one doesn't show it as nice as I'd like, so I have another diagram here to show you the intrinsic factor but this is still one that you should remember. So just to show it a little bit more clearly, we're going to look now at this one, because this one shows a little bit more step by step. What you have here now, once again, is remember we have our factor 12, right? Factor 12 is normally coming from the platelets themselves or from the damaged tissue. Then this now becomes activated, and then the activated factor 12 is going to activate factor 11. Then the activated factor 11 activates factor 9, and in factor 9 is activating factor 10. So we go here 12 to 11 to 9 to 10. If you can remember 12, 11, 9, 10, you have remembered the intrinsic pathway. Okay? So 12, 11, 9, 10. If we go back here now, it should be a little clearer to us now, right? So remember now, we start with 12, right? And in 12, of course, with the platelet factor and the calcium leads to the activation of 11, right? And 11, of course, leads to the activation of the 9, and that's what leads to 10. So you can see this one doesn't have all the steps there, but it has the basic idea, right? So remember, 12, 11, 9, 10, all of them come back to factor 10. Extrinsic pathway, once again, you start with 3 plus calcium plus 7 gives you TF7. That gives you 10. Okay? All right. Now, once you have factor 10 activated, here's what happens next. Factor 10 activates prothrombinase. And of course, you know that's an enzyme. Prothrombinase converts prothrombin to thrombin, which is another enzyme. Normally, when you say pro at the beginning of an enzyme, it means that enzyme is in its inactive form. So prothrombin becomes thrombin, which is now active, and thrombin takes fibrinogen, which you all know is floating around in the blood already, and converts that to fibrin. And fibrin, of course, is what forms the strands that help to hold the clot together, right? So please make sure you have the understanding of this diagram here. These as well, I mean, it's basically the same idea. So if we look at the common pathway using another diagram here, we can see here again, we have factor 10. We have factor 10 being activated. And that, of course, gives us our prothrombinase. And our prothrombinase, remember, converts prothrombin to thrombin. This is still an enzyme. And then that goes thrombin, goes fibrinogen to fibrin, and fibrin makes the clot, right? In addition, thrombin also activates something called factor 13, and 13 reinforces the mesh that makes up the clot, right? So fibrin forms lots of strands, and the factor 13 basically weaves the strands together, joins them together to form a mesh. And in this fibrin mesh catches red blood cells in it. And in that, those red blood cells are shoved down into the wound in the mesh to form the clot, right? So that's all a clot really is. It's a net bag filled with red blood cells shoved into the wound. That's what it fundamentally is. Okay? All right. Before I move on, questions? Any questions? All right. I guess we're good to go. Let's continue. So we can jump from there and I'm going into the cardiovascular system. Now, as you know, we don't have a lot of time. So some of the stuff I am going to go through relatively quickly because I mean, let's be honest, some of this you should already know, but let's make sure. So this, of course, is uh, slightly more complicated probably than the one you got at CSEC. Look at the heart, okay? Uh, Let me run through the basic parts real quick. Then I'll remind you about how blood flows through the heart before I move on, right? Um, First of all, I'm assuming everybody remembers why you have red and blue arrows, right? If not, please let me know. All right, good.
Don't remember.
Okay. Blue, everywhere, when you get a diagram of the heart, generally speaking, if you see blue, that means the blood is deoxygenated, or we just say deox blood, right? Wherever you see red arrows, it normally means the blood is oxygenated. That is where there's a difference, red and blue. Humans are set of a double circulatory system because blood enters the heart twice. The first time blood enters the heart, that blood is deoxygenated. So you can see here, blood is entering the heart via the inferior and superior vena cava. Okay? The superior vena cava is taking blood from your trunk, which is of course basically is your chest, arms, neck, head, bringing that blood back to the heart. Then your inferior vena cava is carrying blood from the abdomen, legs, genitals, and bringing that blood back to the heart. So that's why you need two of them. So one takes blood from the top, one takes blood from the bottom. They both carry that blood, deoxygenated blood, back to the heart. So that means that they both have to be veins. So your vena cava are veins. They're the biggest veins you'll find in the body. And both of them dump blood into your right atrium. Okay? So the first time blood enters your heart, it is deoxygenated and it goes in via the right atrium. Okay? Now, once that deoxygenated blood enters the right atrium, it fills the right atrium, right? Now, I'm going to mention something you may not have learned at CSEC. Um, that is to do with this one here. What I'm going to do is I'm just going to circle the ones that you may not know. Okay? So right here in your right atrium, you have this thing here called the coronary sinus. Coronary sinus is where deoxygenated blood from your coronary system is coming back into the heart. You must have heard people talk about coronary heart disease. Okay? When the heart itself has problems, that's with its circulation, that's coronary circulation. So the coronary system feeds the heart muscle with blood and glucose and all the good things it needs. And then that used blood comes back now to the heart chambers via your coronary sinus. So that now means blood is entering the right atrium from the inferior vena cava, superior vena cava, and coronary sinus. And all this blood is deoxygenated. Okay? Now I want to leave the fossa ovalis and the other ones for a little bit. Let me just talk through now the flow of blood through the heart. Okay? Once your coronary artery, your right atrium is nice and filled with blood, it will contract, right? And it will then force blood past a valve called the tricuspid valve, right, into the right ventricle. So remember now, I'm telling you all things you should know already, right? Note it's called a tricuspid valve because that's three cusps. The cusp is basically a section of the valve. Here we have one, two, and three pieces of it. So one, two, three cusps. It's a tricuspid valve. It's a three-segment valve. So blood gets pushed in through the tricuspid valve into your right atrium. Okay? Now, within the right atrium itself, the walls are a little, well, significantly thicker than the walls of the right atrium. They're much more muscular in comparison. Because now the right ventricle has to push blood out of the heart. Remember, blood is entering the heart twice, which means it has to leave twice. So this is the first time blood is leaving the heart now via the right atrium. When the right atrium contracts, it forces blood up now into something called the pulmonary trunk. So right ventricle goes to pulmonary trunk. Now the pulmonary trunk goes and splits into your pulmonary arteries. So even if they didn't say pulmonary trunk at CSEC, they must have said pulmonary arteries, right? So we have right pulmonary arteries and left pulmonary arteries. There's more than one of them, okay? Now these arteries are going to take the blood and carry that blood to your lungs. And of course, at that point, that's when you pick up oxygen, okay? Now, in case you've forgotten, pulmonary arteries are very unique. They are the only arteries in the body that carry deoxygenated blood. All of your body's other arteries, the blood is going to be oxygenated. Only pulmonary arteries, right? Now, once the blood goes to the lungs and gets all the lovely oxygen it can, it's going to come back now to the heart. This is now the second time it comes in. So remember, it came in first, right atrium, deoxygenated. So no surprise, it comes in now. Next time, it is now oxygenated, and the blood comes in via the left atrium. Okay? So, and it does that via pulmonary veins. You have the left pulmonary veins and right pulmonary veins, which I can't find right now on this diagram. But anyway, so left pulmonary veins, right pulmonary veins. Why? Because you have a left lung and a right lung. So this carries blood back now to the left atrium. And the pulmonary veins are special because they're the only veins that carry oxygenated blood. All other veins, the blood is deoxygenated. So like the superior and inferior vena cava, that blood, of course, is deoxygenated, as you know, right? So once the blood now coming from your lungs enters the left atrium via your pulmonary veins, the special ones, they get pumped now into your left ventricle via another valve called the bicuspid valve, also known, of course, as your mitral valve. I prefer bicuspid because if you have tricuspid on the other side, why change it, right? So the bicuspid valve is called thus because it has two parts this time instead of one. Hence, bi. Remember, tricuspid valve has three parts. Bicuspid valve has two parts. So blood now from left atrium gets pumped into the left ventricle when the atria contracts. And the left ventricle now has the thickest walls of any part of the heart. Why? Because this one now has to pump blood to the entirety of the body. Remember now, on the right side, the right ventricle is just pumping blood to the lungs. And the lungs are right beside it, basically, the heart. The left ventricle has to pump blood pretty much all around the body. So it has much thicker, heavier walls. So now, left ventricle contracts and pushes blood up into our aorta. And the aorta, of course, is the largest artery in the body. So the vena cava are the largest veins, and they carry blood to the heart. Aorta is the largest artery, carries blood away from the heart, right? As a matter of fact, if you want to say one thing about arteries and veins that is always correct, that is what you're going to say. Arteries always carry blood away from the heart, whereas veins always carry blood to the heart. That is always the case, okay? All right. Any questions here? Hopefully that helps to remind us of how this thing works. Anything you want me to touch on again before I look at the stuff you might not know yet?
Sir,
Yes.
Yes, just to recap, you said um the left atrium pushes blood via the pulmonary veins and
Oh, no, no, no,
then
no, 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 no. The left atrium receives blood from the pulmonary veins and then pushes it um into the left ventricle through the bicuspid valves. So
right,
right,
and then it from the bicuspid valve it goes to
left
where?
ventricle. So it passes through the bicuspid valves into the left ventricle. And then when the left ventricle contracts, that blood goes into the aortas. And from there, in the aorta, I should say, and from there it goes to the rest of the body.
Okay, so
All right. So
thank
just to remind of the order. We go vena cava first, and of course coronary sinus, into right atrium, then through tricuspid valves into right ventricle, then through pulmonary trunk into pulmonary arteries, then from pulmonary arteries to right, left and right, of course, to lungs left and right. That's the deoxygenated blood. Then coming back now from the lungs as oxygenated, we we do left and right pulmonary veins into the left atrium. Then from the left atrium through the bicuspid valves, we go into the left ventricles. And then from the left ventricles, we go into the aorta, which carries blood to the rest of the body. Okay? So that's my C-sec revision. Um, any other questions?
Sir, could you repeat for the coronary sinus, where does it take
That
the blood? You said?
that blood is coming from the coronary circulation. Coronary circulation is what gives blood to the heart tissue. So the heart muscle can't the heart can't absorb oxygen from the blood passing to the atria and the ventricles, right? So that blood has to go into the heart through the coronary circulation. So that used blood goes back to the heart via the coronary sinus. All right. Let me get rid of all of this. All right. So that said, now let's talk about the structures that you may not have had mentioned to you at CSET level. Let's start with this one here. Fossa ovalis. I don't like orange. Fossa ovalis. Fossa ovalis is called the oval window. This structure here connects the right atrium and the left atrium. When you were a fetus in your mother's womb, you didn't breathe via your lungs. You got oxygen from the umbilical cord, and that oxygenated blood passed through your heart to go to the rest of your body, right? But of course, that blood didn't need to go to the lungs to get oxygenated. So only a small amount of blood needed to go to the lungs just to keep the lung tissue functional. The rest of it could go straight to your body. So the fossa ovalis here, which should have been open when you were a fetus, allowed blood from the right atrium to flow right over into the left atrium and from there to the vena cava and then get pumped to the rest of your body. Now, when you are born, the oval window, that fossa ovalis, is supposed to squeeze shut and eventually it should fuse over. And what all it should have really is just a little depression where it is supposed to be. That's what normally happens. In some babies, it doesn't seal over properly, and what you have is it leaks. So that now means that some babies they're born with heart problems because blood from the right side is constantly leaking over to the left side. So you have deoxygenated blood mixing with oxygenated blood. And this gives them something called blue baby syndrome. They're listless. They don't really cry very much. And if they're light skinned, that their skin looks kind of grayish, you know, that's blue baby syndrome. And when that happens, of course, a surgeon has to go in and so the fossa ovalis shut. Or for most of them, eventually that baby will die. Okay? So fossa ovalis, probably a very important structure you've never heard of. Then now, something else you might not have heard of are pectinate muscles. These are special ridges of muscle inside the atria, and they help to tension the atrium and help squeeze out the blood when the atrium is contracting. Okay? So they're assistant muscles basically. So they don't, so the entire wall of the right atrium isn't very thick, but it does have these little bands of muscle just to help out, right? You can imagine when you're running for your life, or you're running a marathon or a race or something, and your heart needs to be pumping faster, and it needs to be, so the right atrium has to be squeezing blood out rapidly. The pectinate muscles are very helpful for squeezing out that blood, right? Then now you have this interesting area called the conus arteriosus. This, of course, is where you have the right ventricle meeting the trunk, the pulmonary trunk. It's called a conus because the area is sort of cone shape. You know, it kind of looks like a cone if you really squint and turn your head. So it's the conus arteriosus. Then now, here's another one you might not know, which is your papillary muscle. Now, these muscles are coming off of the walls of the ventricles. They look like little lumps. And attached to these papillary muscles, we have strings. And these strings are called the chordae tendineae. If you've ever heard the term heartstring, you must have heard us, somebody says, tell you a sad story or sing a sad song. Say, boy, it pulls on your heartstring. So the chordae tendineae are your actual heartstrings. Your heart actually does have them, right? What this has to do with you being sad, I have no idea, but they're there. The papillary muscles are attached to these heartstrings, these chordae tendineae, and in those chordae tendineae are attached to the cusps. Remember, the cusps are the flaps here of your valve. And the papillary muscle, by pulling on the chordae tendineae, are going to pull on those flaps. And depending on how much tension the papillary muscles put on the flaps, it can help to open them or help to close them, right? So the papillary muscles help to move the valves. So when you want to push blood in, they can help. When you want to keep the valves closed, they can help, right? So between the papillary muscles and the chordae tendineae, they assist the valves function, right? Then now, on the other side, the aortic arch here, we're looking at the major blood vessels coming off of the aortic arch. So we have here the brachiocephalic trunk, and y'all did muscles at AMP1. So you should know what brachial refers to, as in the brachial area, and cephalic, of course, as in head. So the brachiocephalic trunk, of course, is carrying blood to head and arms, right? Well, then we have here the left common carotid artery. This one's going to the neck. Then we have the left subclavian artery. Sub, of course, is below. Clavian is clavicle. So that one is going to the left side of your body below the clavicle. That one normally goes into the arm, right? So those are the three major blood vessels coming off of the aortic arch. So you need to remember those as well. Then there is another one you may not have heard of before. The ligamentum arteriosum. As you can see here, it's a ligament that attaches the pulmonary trunk and the aortic arch together. So it helps to keep everything stable as the heart is pumping, right? Then going down, going down, going down. I think we all know what a septum is already. Septum is going to separate, right? So in this case here, you have an interatrial septum. So you have a septum, a wall of muscle that separates the right atrium from the left atrium. And of course, you have the interventricular septum, which separates the right ventricle from the left ventricle. Okay? Now, this one here is another one you may not have heard of before. The trabeculae carneae. The purpose of the trabeculae carneae is to allow the ventricles to contract fully without creating suction. What do I mean by that? If you've ever blown into a balloon, you know when you're young, you blow up the balloon, you let the air out, you blow it up, you let it out, and you get spit in the balloon, and you put it down, and you take it out back, and the balloon just sticks together and doesn't want to separate because the membrane of the balloon is wet and smooth, so it wants to stick to itself. The surface of the heart is very, very smooth, right? So it sticks together worse than two sheets of plastic. And so in order to prevent the heart muscle from sticking to itself, you have these trabeculae carneae, which are these irregularities inside the chamber. What that means is that only a small amount of the heart tissue is touching when the ventricles contract. So there's not enough suction to hold the ventricles together, right? So it stops the ventricles from sticking to each other when your ventricles contract. Okay? The moderator band here assists in the contraction, so sending signals of contraction to the ventricles. All right? All right. Questions, comments before I move on from here? Any of the new ones that you're not sure about?
So you said send signal of
Into the
contraction.
ventricles to help with tensioning and contraction. But I'll mention it a little bit later on, right? All right. So hopefully the heart, this you remember this. This here is just showing a general location of the heart. I mean, just read through it, it's not a big deal. Now, this is giving you basics of the heart, um, basic physical characteristics of the heart. Keep in mind, this is average, okay? Bigger people tend to have bigger hearts, right? Athletes. Well, let me ask you, do you think all athletes will have bigger hearts? Or let me make it easier. Which one do you think will have the biggest heart? Um, a professional weightlifter or a sprinter? Who do you think has the bigger heart?
I'd
Maybe
say
this.
a sprinter.
And
That's what I think.
you would be correct. Weightlifters will not have large hearts because, of course, they don't need them. I mean, how long you lifting the weight for? A couple seconds? Whereas a sprinter has to be going on for a good little while. So the more cardiovascular exercise your sport requires, the bigger and stronger your heart will tend to be, right? So the figures you're seeing here, this is for your average person. You know, I don't know if there are any athletes in here, but if you're an athlete, then your heart is probably going to be bigger than this, right? But on average, and of course, men tend to have larger hearts than women as well, right? As Emma says, approximately 12 and a half centimeters from the base to the apex. Now, apex and base are always annoying terms. Here's the reason why. When we go back and look at the heart, this is in situ. In situ, of course, means in its natural space. So this is where the heart would sit in you. The apex is the tip of the heart, and as you can see, it is pointing down and to the left. Normally, apex means the top of something. Where the heart is concerned, the apex is the bottom tip, which means, of course, the base is up here, which seems ridiculous, right? Just remember, please, the apex of the heart is the tip of the heart where it narrows, and the base is the top with all the blood vessels coming out and going in, right? So from base to apex, that's 12 and a half centimeters. The heart weighs around 250, 350 grams. No one cares about ounces. 250 to 350 grams. That's basically the weight of a small bottle of soda. think a small bottle of soda is about 250 mils, right? Then now, we know the heart itself, mid-sagittal section through the trunk, does not divide the heart into two equal halves because the heart is not bilaterally symmetrical. I'm not going to spend too much time on this because I know you all know this already from AMP1. Remember, the heart is not bilaterally symmetrical. You cannot cut the heart in two when you run a line down the body, right? Into two equal halves, I should say, right? It is very, very much asymmetrical. This is looking, of course, at the general features of the external heart. I know you all know these already, so we won't spend too much time on it. Now, pericardium. Anybody in here eat heart? Anybody ever cook a heart? Like you get like a goat heart or something?
Yes,
No,
sir. Or
sir.
liver. Or chicken
One
liver.
person. Wow. First of all, if you know how to cook heart, heart is really nice. It tastes like a mineral roast beef. Normally, you braise it for a nice long time, or if you have one of those nice crock pot slow cookers, just put it in there, leave it for 12 hours, with some spices in there. Nice, nice. Anyway, um, but most organs, as you know, are going to have a membrane around them, okay, which helps to protect them. The name of the membrane is going to be associated with the organ. The membrane around the heart, of course, is called the pericardial membrane or the pericardium, right? Inside you have the pericardial cavity, which is going to have lubricating fluid, okay? The, the pericardium now has two main parts. You have what is called the visceral pericardium. This is the part that directly touches the heart itself. And you have the parietal pericardium, which is what they're lifting up here to show you, which doesn't touch the heart directly itself, but it does touch the chamber the heart is in, which is the pericardial cavity, right? So here, this is what it's showing you here. Here's the pericardial cavity. Here's the serous membrane. Here's your visceral pericardium. Here's your parietal pericardium, right? Here's your heart in between. Sorry, here's your heart, I should say. And of course, you have your lubricating fluid between the visceral and parietal pericardium, right? Once again, that's MP1 stuff. We're going to spend too much time on it. So just review this to remind us of what you did in AMP1. This too. So epicardium is the outermost layer of the heart. Myocardium now is the muscle part, that's the middle part. And the endocardium now is the inner part where blood is in direct contact with. Okay? So the epicardium is basically the visceral pericardium. Right? So the epicardium sits directly on the heart. That's the visceral part of the pericardium. Then below that, you have the muscular layer. That's the myocardium. That's the pumping part. And then inside the heart itself now, touching the blood, you have the endocardium. And this is made up of simple squamous epithelial cells, right? All of this is still AMP1 stuff, right? So I'm going to just jump ahead now, and I want to show you this image here because when you think about the heart muscle and how it's arranged, people rarely think it looks this way. So look at the way you have the arrangement of the muscles in your atria. This is kind of what you'd expect. You see cylinders of muscle running through the atrial walls. So when the atria contracts, it contracts inwards like this, right? So that's sort of what you expect. But look at what is going on with the ventricles. You don't have these circles of muscle in the ventricles. Instead of it, you have these Mobius strip-like bands of muscle running through the ventricles. And here's why this is important. Do you see the way the muscle twists through the ventricles? When your ventricles are contracting, they don't just squeeze down like you squeezing on a rubber ball. They squeeze and twist at the same time. So when the ventricles are contracting, it's more like when you're wringing out some clothes. I don't know if anybody in here still wash clothes by hand. I know someone on the tools touch. But for those who, I mean, I assume you wash underwear by hand. You have to wring them up before you hang them up. So when you're thinking about how the ventricles contract, think about that. You're twisting and as you're wringing, you're twisting and wringing. And this twisting is very important because the twisting allows you to squeeze more blood out than otherwise you could. Right? So there's a reason why you have the muscles is in this band, twisting band. Right? So please remember that. The ventricle muscle has a much different arrangement from the atrial muscle for that reason. Ventricles squeeze down like you're squeezing on a rubber ball in your hand. Atria squeeze down like you're squeezing on a rubber ball in your hand. Ventricles twist and squeeze, right? All right. I mentioned the simple squamous epithelium for the endocardium already. I won't go back to it right now. Now, for the external features of the heart, let's talk about the ones you may not have done at CSEC, right? I'm assuming we did vena cava already. You know right atrium already, you know left atrium already. You know all of those pulmonary veins. We did all of those, right? Now, the ones that you may not have touched on are the coronary sulcus and the anterior interventricular sulcus, okay? A sulcus is a groove, right? It's a depression basically running down the heart. And the reason these sulci are important is because they allow blood vessels to run in them, okay? So you have the coronary sulcus, which is a depression between the atria and the ventricles. So here's the atrium, here's a ventricle, here's a coronary sulcus. This is the area of one coronary sulcus. Here's the area of the next one. They allow the coronary arteries to run through there, okay? Then now you have an interventricular sulcus. You don't have to ask where that runs because it tells you. Interventricular between the ventricles. So you know this area here is where you'll have your interventricular sulcus. As I said, the name tell you exactly where it is. Okay. Now tomorrow you will be dissecting a heart. You'll be doing one drawing of external characteristics of the heart, one of the internal characteristics. For the external one, I'm expecting you to label the sulci as well. So keep in mind from now where they're located, right? Now, this is just the wording to what I just said. So you can read through that in case you forgot what I've said, right? That said, I mentioned those already. I won't mention them again. Did I mention ductus arteriosus? No, I don't think so. No. Okay. Ductus arteriosus. The ductus arteriosus here. Remember, I mentioned the foramen ovale earlier, right? Or oval foramen. And I said to you, the foramen ovale connects the right atrium and the left atrium when you're in the womb. So it allows you to send blood from the right side of the heart straight to the left side of the heart, bypassing the lungs. The ductus arteriosus is also something that you have in the fetal stage. And if you notice here, let me make it a little bigger so it's a little easier to spot. The ductus arteriosus is right here. So this is the pulmonary artery, and it merges here with the aorta. What this means is that blood coming from the right side, right ventricle, goes straight into the aorta. And of course, some of it does go to the lungs because the lungs will need oxygen too at this stage. But you will also see it's going straight into the aorta. Okay. So between the foramen oval, which moves oxygenated blood into the left side, and which eventually goes into the aorta, you also have blood from the pulmonary trunk via the ductus arterius also going into the aorta. Remember now, while we're doing this, the blood is already oxygenated coming from the umbilical cord into the heart, right? From the vena cava. So that means that blood can go straight to the body. Hence, foramen ovale, hence ductus arteriosus, okay? So remember, those are only found in the fetal stage of development. Obviously, you need to be able to label this diagram, right? And I will leave you to butter with that one, right? Now, I think I went through most of the internals of the heart already, so we don't need to run back to this again. See, is there anything I didn't mention? Mention all of those and all of that. So I mentioned pectinate muscle already too. Good. Chambers, awesome. Ventricles, we mentioned those. I mentioned the moderator band in passing, right? I just says it transmits impulses from the right bundle branches. I'll explain what that means when I go to electrical signals in the heart. We already know valves prevent backflow of blood. That's what they're for. We know that already from CSEC. We did the valves here. Yeah, we're not doing too bad. All right. So this is just showing you here now in a little bit more detail what the valves look like. Okay. So let me stop here for a second. So this here is the bicuspid valve. Okay. It has two parts to it. One side, the other side. It sort of looks like a cup without a bottom, right? And then, of course, here are choroidae tendineae attached to our papillary muscle. Remember what this is for? To tension the valves to be able to keep them closed when they should be closed, right? Then now we have, of course, this lovely diagram here that shows you how this works. If you notice here, when the papillary muscles relax, the valves pop open, right? When you tension the papillary muscles, they pull on the choroidae tendineae, which are attached to the valves, and that pulls the valves closed and prevents the backflow of blood, right? So remember now, papillary muscles tension the valves, keep them closed. All right. Tricuspid valve, bicuspid valve, we did that already. Oh, semilunar valves, we didn't mention this one. In the pulmonary arteries, you will have what are called the pulmonary semilunar valves. And their function, of course, is to stop the backflow of blood that has left the heart and gone into the pulmonary arteries on their way to the lungs. Okay? Remember, arteries do not normally carry valves. But in this case here, you have a pulmonary valve. All right? Sorry, this is in the pulmonary trunk, I should say. So the pulmonary valves in the pulmonary trunk, remember the trunk splits into the pulmonary arteries, right? So this stops blood from re-entering the heart. Oh, right? It's one of the few arteries you'll find with any sort of valve. Then on the other side now, you have the aortic valves, which of course are going to be in the aorta, between the aorta and your left ventricle. And of course, it does the same thing. It stops the backflow of blood into the heart, right? That, of course, means blood flows to the heart in one direction only. Once blood leaves the heart, it has to go all the way to the circulation to get back to it, right? Now, this is just showing you a section, a transverse section showing the valves and where they're positioned, okay? Can't spend too much time on this one. But you can see the aortic valve here has 3 sections, and the pulmonary valve has 3 sections as well, okay? But they're not tricuspid valves, okay? Please remember that. They are not tricuspid valves. You don't see any choroidal tendon or anything like that on these valves. These are just ordinary vascular valves. Tricuspid and bicuspid are special. All right. So we're already in through tricuspid and bicuspid valve for the most part. So we're not going to spend too much time on those either. Here we go. Foramen ovale and the fetal heart. So we spoke about that already. We can jump ahead. Here's another lovely diagram for you to make sure you can label. All right. Now I mentioned already that the circulatory system in mammals, like ourselves, is a double system. And I remember saying to you, because it was not long ago, like half an hour ago, that it's a double system because blood flows through the heart twice. Once as oxygenated blood and once as deoxygenated blood, right? So remember now, first circuit goes blood from the body, enters the heart, then from there, it goes to the lungs, then back to the heart. So we can say heart, lungs, heart, and that is the pulmonary circuit. Heart, lungs, heart as pulmonary. Then now the next circuit, which sends blood to the whole body, is the systemic circuit. So blood, of course, from the pulmonary circuit enters the left side of the heart. So we'll start with the heart again, and we go heart, then to the whole body, then back to the heart via the vena cava. So systemic circulation is heart, body, heart. Pulmonary circulation, heart, lungs, heart. Please don't mix them up, okay? All right. This here is just giving you a list of some of the structures that you will find associated with each circuit. And I'll leave you to review those yourself, right? All right. Now, before I move on to coronary circulation, any questions, anything I should clarify before I move on? If I'm going too fast, let me know, please. But we do have a limited amount of time, as you know. All right, everybody's good so far? Okay, coronary circulation. Now, remember what I said to you about the coronary circulation. Its job is to supply the heart muscle itself with blood, right? So, that said, let's talk about the major vessels associated with your coronary circulation. The first one that you will be aware of will be your coronary arteries. Now, the reason you're going to be aware of these is because half of the time when you hear people have heart problems, they say that the problem is their coronary system, right? Now, why is that? The blood going into the coronary system, right, is blood that has been oxygenated and goes from the heart directly into the coronary system. And that blood tends to be heavy with fats. Okay? So that blood tends to have a lot of fatty acids and glycerol bound to lipoproteins in there, which for the heart is good because the heart needs this stuff for energy. Okay? That blood is really rich in nutrients. Problem, of course, is that if you have a diet that is very, very high in the saturated fats, etc., it is easier for these things to form plaque within the coronary arteries. because of how rich that blood is. So that's why normally when you hear coronary heart disease, they're talking basically about blockages of the coronary arteries that feed the heart itself with blood. Now, where do the coronary arteries begin? It mentions that the left and right coronary arteries originate at the base of the ascending aorta, at the aortic sinuses. Now, what is the ascending aorta? It is literally where the aorta leaves the ventricle. It goes up first, then it curves over. So this is the ascending aorta here. And then this part here is the aortic arch, right? So your coronary arteries are coming from basically where the aorta meets the ventricle. So right here. So that now means that the blood that is coming straight from the ventricle, right, is going to go to the heart first. The heart is the first structure to get fed blood, which of course makes sense because it is the one that is doing all the pumping and all the work to move the blood around, so it should get fed first, right? Now, this here is looking now at where the aorta meets the left ventricle. Here we have our aortic valves, okay? Now, inside the valves, you notice that the valves kind of like a cup, right? Inside the valves, we have a little space because, cups have spaces. And you see it's referred to as the aortic sinuses. So here's what I want you to imagine. Notice here inside the cups, there's a little pore, a little hole. And this hole now is where you have the coronary arteries beginning. So blood entering these little orifices go into the coronary arteries. So we have a right coronary artery, left coronary artery in our little cup. Remember, our little cup is our valve, right? So here's what I want you to imagine now. Imagine blood getting pumped up out of your left ventricle, right? So the ventricle squeezes and pushes that blood up and out. When the ventricle relaxes, blood falls right back. And because the valves are there, and the valves are like cups, the valves catch the blood and stop the blood from going back into your ventricle. But because you have little holes in your cup, of course, if there's a hole in the cup, blood leak, something will leak out. So the blood flowing back now into the valves gets squeezed into the coronary arteries. And from there, of course, this blood can go into the heart muscle, right? So every time the ventricles relax, blood gets pushed into the coronary arteries, right? Why? Remember, the ventricles relax, blood falls back down into our little cups and then goes into the holes, and from there it goes into the coronary arteries. So in this case here, it is a relaxation of the ventricles that helps to move blood into the coronary arteries. And if you think about it, it makes sense. When the heart muscle is relaxed, it's easier for blood to flow into the vessels on the surface of it, right? If you think about it, because they won't be squeezed down by the heart. All right. If that doesn't make sense to you, please let me know, right? So remember now, as it says, the elastic rebound forces blood through coronary arteries in between contractions. So this is what I was just explaining. As the ventricle relaxes, blood coming back down pushes its way into the coronary arteries, right? Now, from the coronary arteries, we're going to see what branches off. So if we follow our right coronary artery here, you can see it runs down our septum, our sulcus. So the atrioventricular sulcus. So here it is, right coronary artery. And then branching off of the right coronary artery now, you're going to have the posterior interventricular artery and the right marginal artery. On the left side now, the left coronary artery runs down now, and then you get this circumflex artery and interventricular artery. So this one branches off and runs down into the interventricular sulcus. Okay. Now, I know it's obvious, but let me state it clearly. The ones in bold are going to be important. All right. So keep that in mind for me, please, right? So as we go along, the ones you see here in bold are the ones you're expected to remember. So here it mentions that the marginal arteries and that they extend from the right coronary arteries over the surface of the right ventricle. Right? So you can imagine how easy it would be for me to write multiple choice questions, just basically asking, hey, where do these arteries run to or from? Right? So as you're going along, that is what I want you to internalize, to remember. Remember where the arteries are coming from. Remember where they're branching off from, where they're going to, right? Then now, when you get to the veins, you're going to do the same thing. All right, I don't have time to go back and say this is the artery, that's the artery. Just read through this for me, right? I do want to mention one, which is the anterior interventricular artery, because it forms things called anastomosis. This word you may not have heard before. Basically, anastomosis refers to an area where multiple arteries merge together to form a bigger artery going into one structure. Okay? So it mentions here that you have an anterior interventricular artery that forms an anastomosis with the posterior interventricular artery. All that means that these two arteries merge together. Okay? So anastomosis, when you see the word, arteries, two or more, merging together. That's all it is. So don't panic when you see the word. Just combining arteries. That's all it is, right? So please remember, go through, make sure you remember the important ones, the coronary arteries, where they're coming from, where they're going to, posterior interventricular, marginal arteries, anterior interventricular, circumflex, where they're coming from, where are they going to, right? Same thing with the veins, right? So coronary sinus, remember it leads into your right atrium. So what feeds the coronary sinus? Of course, the cardiac veins, greater cardiac vein, middle cardiac vein, small cardiac veins. Once again, where are they coming from? Where are they going to? I mean, most of them will empty into the coronary sinus still, right? So please make sure that you internalize this information. There will definitely be questions from this section on your first in-course test, right? Definitely questions from coronary circulation will be coming on your first in-course test. This diagram here summarizes the major vessels you'll find in the circulation, coronary circulation. Okay. All right. Everybody understand what I'm saying? Remember, it's a must. Questions will be coming from here. Just remember, where is it coming from? Where is it going to? You can remember that. Should be fine. All right. That said, let's talk about heart sounds. Everybody knows heartbeat, and everybody knows what the heartbeat is supposed to sound like, right? Now, generally speaking, if you ask somebody what a heart sounds like, almost everybody will tell you boop, 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 boop. Unfortunately, that is not how we classify heartbeats. You can't tell a doctor the heart sounds boop, 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 boop. Instead, you have to use the terms lob and dop, right? Now, if you've ever listened to the heart, you know it doesn't really sound like lob or dop, but we have to pretend that it does. So basically, whether it's boop boop, boop boop, or lob dop, we can agree that it's two sounds. You either get the boop or the boop, or the lob and the dop, right? Two sounds. So where are these two sounds coming from? The lob is what the sound you hear from the lobe occurs after the atrioventricular valves close and, of course, blood hits the valves. Okay? The atrioventricular valves, of course, are the valves between the atria and the ventricles, which means I'm talking about the tricuspid valves and the bicuspid valves. Okay? Generally speaking, the right atria and the left atria contract at the exact same time and force blood into the ventricles, right and left, at the same time. Okay? And when after that happens, the ventricles, right and left, are going to contract at the same time, which means that the tricuspid valve and the bicuspid valve have to be closed at the same time, right? So blood hitting both those valves will contribute to that first sound, the lob sound, which is the loudest of the two sounds. Okay? So AV valves slamming shut, blood smacking into them, lob sound. That's it. Okay? Remember, um, um, atria contract at the same time, loading both ventricles. Both ventricles contract at the same time, closing the AV valves at the same time, giving you the sound. Blood then rushes out of the ventricles. The right ventricle, of course, sends blood to the pulmonary trunk through the pulmonary valve. On the left side, of course, you have blood from the left ventricle going up past the aortic valves. Now, when the ventricles relax, they're no longer squeezing blood out. The blood in the pulmonary trunk falls back and smacks into the pulmonary valve. Blood from the aortic side, the blood in the aorta falls back, smacks into the aortic valve, and you get sounds from that. Okay? So the slamming shut of the of those two valves, we call those the semilunar valves. Remember, please. So the pulmonary valve is a semilunar valve. The aortic valve is a semilunar valve. So blood hitting the semilunar valves gives us the dop sound, which is the second sound, okay? So lob, remember, is blood from the ventricles smacking into the AV valves, all right? And the dop sound is blood in the smacking into the semilunar valves, right? So those are your two heart sounds. Now, that said, I want to start talking now about how the heart coordinates the beating, okay? This, of course, is referring to now the conducting system. So this is, remember I said to you, the two atria contract at the same time, then the valve is shut, and then they relax, and then the two ventricles contract at the same time. So there has to be a mechanism coordinating so that the atria close at the same time and the ventricles do their thing at the right time. That is now looking at your conducting system. So we're looking at what coordinates what is going on in the heart during your normal heartbeat, right? So that's the conducting system we're going to be talking about, right? This here is looking at the microscopic anatomy of the heart. This stuff you need at AMP1, I'm not going to spend too much time on it. You should already know, for example, that your heart muscle cells tend to be relatively short and they tend to be bifurcated. They tend to be in two parts. So they split like a Y. And they have the intercalated discs. And you know those help to transmit impulses quickly through heart muscle. All this will sound familiar to you from AMP1. You already know that the heart muscle cells can be multinucleate. They can have up to two nuclei. They're striated. All of this stuff should sound familiar to you, right? So we're not going to spend too much time on it. Um, this I'm not sure if you learned at AMP1. It mentions here, heart muscle is stimulated by nerves and is self-excitable. So this means that heart muscle is what we call myo muscle genesis, right? Myogenic. Heart muscle can generate its own impulses to contract, which is kind of interesting because what it means is that it does not need signals from the brain to contract. So you can actually cut out the heart out of an animal and the heart will continue to beat for a while, even if it's not attached to the body. I don't know if you've ever seen movies where people get their hearts ripped out and somebody's holding the heart and the heart is beating. Um, but that's actually a thing. The Maya who used to sacrifice people to their gods, that's something they'd do. They'd carry them up to the top of the pyramids and strap them down over the altar, and they'd use an obsidian knife, and the priest would cut open the heart of the captured slave and rip out the heart and lift the heart up to show everybody the heart is still beating in their hand. As a real thing that actually did happen, right? Now, remember, it can continue to beat for a while because it is myogenic, right? It generates its own impulses to contract. Now, it mentions here that heart muscle has a long refractory period. What does this mean? It means, of course, as you know from AMP1, when you did muscle contraction, it, there's a long period between when the heart receives one signal to contract and then before it can respond to another signal to contract. Okay? Now, why is this important? It says here it has a refractory period of about 250 milliseconds. This is saying that the heart cannot contract more than 4 times a second. 4 times a second is a ridiculously fast speed, right? Now, why do you want to slow down the rate of heart contraction? You think, hey, the faster the heart contracts, the more blood flow. That's not good, man, but only up to a point. If the heart is contracting too rapidly, it will not be able to finish one contraction and properly empty the chambers before it starts another contraction. And that, of course, is going to cause problems. That is why you need to have this relatively long refractory period to give one contraction enough time to empty whichever chamber is contracting and then allow for the chamber to fill with blood again before the next contraction occurs. Okay? Now, autorhythmicity cells will control now the heart rhythm, right? Heart rhythm is extremely important. The heart, I always tell students, the heart is the hardest working organ in the body that doesn't want to work. Here's what I mean by that. It works real hard until you get to give it an excuse not to. The moment it gets an excuse, even no matter how small, to stop, it just stops. Right? So the heart is contracting in a certain rhythm. If the rhythm is interfered with, the heart will simply beat out of rhythm and then the whole process just stops, and that is your heart attack. Right? This is the reason if you punch somebody in the chest at the wrong time, you can actually disrupt the beating of the heart and kill them. So people have died from being punched in the chest. All right. People have fallen and hit their chest and died from the fall. So yeah, the rhythmicity is a very serious problem, and maintaining the rhythm is required to maintain the heartbeat. If the rhythm goes out of sync, the heart will just stop. Right? Well, strongly enough out of sync. To maintain the rhythm, we have special types of cells called pacemaker cells. And I want to talk about the pacemaker cells next. Now, before I do that, I've been going on for almost 2 hours. So I want to take a very brief break, a 1 hour, a 10-minute break, and then we'll come back, okay? I don't want this video to be too big, so I want it in two parts. Before I go on the break, any questions?
No, sir.
All right. So remember, 10 minutes. That means you come back at 7 for the last hour, okay?